The overall aim of this procedure is to demonstrate a method of monitoring the calcium signals at both the cellular and whole plant level. This is accomplished by first growing seedlings on the MS agar plate or directly within the imaging chamber. The second step is to transfer the seedlings onto an adhesive film or to keep them in the imaging chamber. Next, stimuli are applied to the seedlings to induce calcium signals. Ultimately, live cell luminescence or fluorescence imaging is used to show whole seedling or cytosolic nuclear calcium response respectively to different stimuli.
The main advantage of this technique over existing methods like single plant aquarium assay is that film adhesive seedlings assay is simple, sensitive, and robust. Therefore, this technique can be adapted to the high throughput screening for mutants with altered calcium signal responses. In addition, compared to FRAP-based calcium imaging, using GFP-based genetically encoded calcium indicator avoids complicated microscopic settings and calculations.
This method provides insight into calcium signaling during the stress response in Arabidopsis, but it can also be used in other contexts, such as during development or defense in other plant models.
Demonstrating this method will be Shen-Yi, a student from our lab.
To begin this procedure, sterilize the seeds of Arabidopsis plants expressing aquarin with 10% bleach solution, containing 0.01% Triton 100. Sow the seeds on a square plate containing full-strength MS, 1% sucrose and 1.2% agar. Next, place the plates vertically in a growth chamber after stratification at 4 degrees Celsius for 2 days. Afterward, transfer the seedlings onto a film. Place an adhesive film on top of the 7 to 10 day old seedlings growing on the plate. Gently push the film by hand to ensure that the seedlings adhere to the film. Then, peel the film gently so that the seedlings remain adhered to the film. Next, incubate the seedlings with the cofactor by placing the adhered seedlings onto a square plate containing 15 milliliters of 2 micrograms per milliliter HCTZ in water. Incubate the seedlings at room temperature for 4 hours to overnight. To prepare for luminescence imaging, take the film out of the HCTZ solution and cut it down the middle to form 2 pieces. Place the 2 pieces of film with the seedlings facing up in 2 different plates. Then, leave the plates in the dark for 5 minutes. To acquire luminescence images, in the dark, place 2 plates next to each other on the stage of the luminescence imaging system. Acquire images immediately upon adding 20 milliliters of stimuli solution to the plates simultaneously. To analyze the luminescence images, choose the same display range for all luminescence images. Crop the ROI and generate the images as JPEG files. Alternatively, export the images as SPE format files and import them into the ImageJ image analysis software. Set the measurements for calculating the mean gray value of ROI. Select the same size of ROI area. Then, measure the mean gray value and present data as bar graphs. To prepare seedlings for confocal imaging, sterilize the seeds of the Arabidopsis plants expressing Ca2+ with 10% bleach solution containing 0.01% Triton. Sow the sterile seeds on a plate containing full-strength MS salts, 1% sucrose and 1.2% agar. Then, place the plates vertically in a growth chamber after stratification at 4 degrees Celsius for 2 days. In the meantime, assemble an imaging chamber with a slide and a cover slip. Place a piece of water-soaked cotton wool on the top of the slide. Then, transfer 1 or 2 5-day-old seedlings from the plate onto the middle of the slide. Then, stick small pieces of clay to each corner of the cover slip and place the cover slip on top of the seedlings to create a gap between the cover slip and slide. After that, connect one end of the polyethylene tube to a 1 milliliter syringe and place the other end of the tube immediately adjacent to the chamber. Hold the tubing in place with tape. To assemble an imaging chamber with a plexiglass chamber, spread a thin layer of silicone grease around 2 polyethylene tubes and press the coated tubes into the channels on each side of the plexiglass chamber. Then, spread a thin layer of silicone grease on the surface of the chamber that contains the tube grooves, and place a cover slip onto the grease to seal one side of the chamber opening. Next, transfer a 5-day-old seedling from the plate into the chamber, and place a piece of cotton wool soaked with water on top of the seedling. Spread the silicone grease onto the other surface of the chamber and press another cover slip on top to seal. Afterward, connect the end of one of the tubes to a 1 milliliter syringe and leave the end of the other tube open. To prepare an imaging chamber with a chambered cover glass, first, sterilize the chambered cover glass with 70% ethanol and leave it in the hood until dry. Add 0.6 milliliters or 0.2 milliliters of full-strength MS medium containing 1% sucrose and 0.5% phytogel into each well of a 2-well chamber or an 8-well chamber respectively. Then, sterilize the seeds and sow them directly on a chambered cover glass containing a thin layer of clear gel and let them grow vertically for 5 days. For image acquisition using a confocal microscope, apply stress stimuli by slowly injecting 200 microliters of 150 millimolar sodium chloride, ice-cold water, or 1 millimolar hydrogen peroxide solution into the chamber just prior to acquiring images. Capture the images immediately after applying the stimulus solution using an inverted confocal laser scanning microscope with a 20x water immersion lens. Collect a time series of images at 4-second intervals with excitation and emission wavelengths of 488 nanometers and 500 to 550 nanometers respectively, and at a pixel resolution of 512 by 512. For image analysis, using Nikon elements, ROIs are drawn around each cell of interest. Total intensity within each ROI is measured over time using the time measurement dialog box. Then, total intensity measurements are exported and processed in data graph. Shown here is the comparison of the spatial temporal calcium response of 10-day seedlings to different stress stimuli. Here is the time series of luminescence images of seedlings subjected to 400 millimolar mannitol and 400 millimolar mannitol plus 1 millimolar copper chloride, 75 millimolar sodium chloride, and 75 millimolar sodium chloride plus 1 millimolar copper chloride, and 1 millimolar hydrogen peroxide or 1 millimolar hydrogen peroxide plus 1 millimolar copper chloride. The upper and lower panels are the luminescence images acquired during the first 40 seconds and second 40 seconds respectively. And here is the bar chart of the average luminescence intensity of seedlings in response to the indicated stress stimuli. Shown here is the cytosolic and nuclear calcium oscillation in response to salt stress. The confocal images show that Ca2+ is expressed in the cytoplasm and nuclei of the root cells and the leaf cells of the transgenic plants expressing Ca2+. This image is a merged image of the red autofluorescence of the chloroplasts and the green fluorescence of Ca2+. These two plots show the salt stress-induced calcium oscillation in the cytosol and nuclei of the root cells.
While attempting this procedure, it's important to remember to use well-grown seedlings.
After watching this video, you should have a good understanding of how to monitor calciodynamics at both cellular and whole plant level.